So this is the last part of Unit 2, and what it deals with, it's called absorption of materials, and what you're looking at is how you get important substances into your blood um, for the process of respiration, basically. So it looks at, first of all, gas exchange, so getting oxygen into your blood and removing carbon dioxide, and then later on we'll look at how you get nutrients, um, sugar, proteins, and so on into your bloodstream to be used by your different organ systems. So we'll start off with what's called alveoli. So just a bit of revision to begin with. So there are two tubes at the back of your throat. One of them leads to your stomach, that's your esophagus, and one of them leads to your lungs, and this is the trachea. So when we talk about food going down the wrong way, it's because it's kind of entered towards the opening of the trachea. So the trachea goes down like so, and then it divides into two, and they are called bronchi as a plural, or bronchus. So the bronchi then become smaller and smaller until they become these little circular things here, which are called alveoli. So that is what we're going to focus on. So the plural is alveolus. Sorry, the plural is alveoli. The singular is alveolus. So this is what it looks like. So you imagine your trachea came down and then divided into bronchi, and then your bronchi became the alveoli. So what's happening here is you've got, this is still within the lungs, and then here are blood vessels which are surrounding the alveoli. So the alveoli are commonly known as air sacs, and they're surrounded by capillaries. So you might remember that capillaries are the smallest types of blood vessels. So you've got air coming in from going from the air around you into the alveoli and then into the blood. But then you've also got a gas, carbon dioxide, going back out into the alveoli to be breathed out, and blood is moving up here. So you can see here what is moving from the alveoli to the capillaries. What do you want to get into your blood? A gas that you need. That is oxygen. And you might know that, see the colour here. The blood is always going to be a kind of shade of red, but here, just to simplify things, to show that the blood has got oxygen in it, it is coloured red, and if it doesn't, it's coloured blue. So oxygen goes into the, and what is moving from the capillaries into the alveoli? What's going back into the alveoli from the blood? That is carbon dioxide. So what you've got here is blood, which is coloured blue, not literally, remember, but that denotes that it doesn't have any oxygen in it. So you've got no oxygen, but you've got carbon dioxide. So the carbon dioxide deposited into the alveoli to be breathed out. Meanwhile, you breathe in, oxygen then goes into your blood, and that gets carried to the heart, which pumps the oxygen around your body. Okay, so carbon dioxide out into alveoli and oxygen into the blood from the alveoli. And here's another little drawing of it here. So you imagine this is your bronchiole. So you get the trachea, bronchus, bronchiole, and then you get down to your alveoli. And you can see you've got the blood vessels surrounding it. So this is where the gas exchange happens. So in order for the alveoli to allow that gas exchange to happen efficiently, so efficient, this is diffusion of gases, diffusion just meaning the movement of something from a higher to a lower concentration. Alveoli have got certain features that allow that to happen, to allow the gases to move easily back and forward. So the first thing here, and this is quite a nice representation of it, there's lots of them. Okay, so you can see this is a very small, very small scale, so that there's millions of these in your lungs, I would presume, don't know the exact number, but if you've got a large number of them, then that gives you a large surface area, which just means there are more places, there's a larger area for the gas exchange to occur, for the oxygen to come in and the carbon dioxide to go out. The second thing is that here, between the alveoli and the blood, you have got thin walls. Okay, so again, a sort of simple thing, I suppose. If the walls were thick, it's going to be more difficult for anything to go across them, so you've got thin walls that allows that to happen. And then, finally, where's the oxygen going to go? where's the carbon dioxide coming from? The blood. So alveoli have also got, they're surrounded by these blood vessels, so they've got a good blood supply. I'll mention this, seeing as it's kind of topical, I suppose. This is what COVID does to someone's lungs. So you can see you've got an area here which is sort of white, and what happens is these little alveoli where gas exchange occurs, they're infected by the virus, and what then happens is you've got an immune response. You've got white blood cells. So white blood cells, um, if you're in S4, you'll have done the previous part. So we looked at white blood cells producing antibodies and producing phagocytes, and it's just the immune system response. If something comes into your body which shouldn't be there, the white blood cells coordinate the response to get rid of that foreign body. As a result of the white blood cells moving in, fluid builds up, and if the fluid builds up within the alveoli, it's more difficult for the oxygen to get in. And this is why you'll hear about hospitals requiring oxygen, and also ventilating people as well, because just their alveoli are filled with fluid and it's more difficult to get the oxygen back and forward, or the oxygen in and the carbon dioxide out. So have a look at this, search on YouTube, GCSE Science Revision Breathing, and this will take you through the whole process with kind of animations rather than just diagrams. So to conclude this session, try this. So press pause and then a wee label drawn of gas exchange in the lungs, and you should have the following: blood, alveoli, oxygen, and so on. You might want to do it with coloured pencils, I suppose, to show the blood. And then after that, just beside it, two features of alveoli which make gas exchange efficient. So do that just now and then press play again, and I'll show you what you should have. So I'd said ask for the trachea and then the bronchus and the alveoli. So you should have this bit here, trachea and then bronchus and then alveoli. And then, don't know how you did it, but you might want to kind of zoom in, I suppose. And then what you'd have is a blood vessel, or you might have more than one, a wee blood vessel, and then the kind of key bit is to show that carbon dioxide is going from blood to alveoli, and then oxygen is going from alveoli into blood. Okay, and then the features of the alveoli, there are the three. So I asked for you to have two of them. So we should have any two of these. So a large number of alveoli, which means a large surface area. They've got thin walls, and also they've got a good blood supply. So that previous session we looked at getting oxygen into the blood and carbon dioxide back out of it, gas exchange. The next section we're going to look at is digestion. So we're going to move on to how you get useful substances into your blood, sources of energy basically, and what your body uses to build up proteins and so on. Before we do that, let's see if we've got the basics of the digestive systems. This is revision from S1 and 2. So press pause and put down numbers 1 to 12 and write down the different parts, see how many you can do. So number 1 is the mouth. 2 might have been a bit tricky. The back of your mouth, you've got 2 is the salivary glands, and saliva is mentioned because it contains enzymes, which we've encountered before, so they start to break down the food in the mouth. 3 is the esophagus, so one of the 2 tubes at the back of your mouth. And then 4, that is the liver. 5, well done if you got this, that is the gallbladder, which produces bile, which helps break down fat. 6 is the stomach. 7, we encountered this before when we were looking at insulin and glucagon. 7 is the pancreas. And then 8 and 9, 8 and 10, easy to get 8 and 10 mixed up. 8 is where the food goes after the stomach, and that is the small intestine. And then number 9, some of you might have had this out, occasionally causes a problem. 9 is the appendix. 10 is the large intestine, and then once all the energy's got out, has been taken out of the food, the waste products are stored in the rectum, which is 11, before finally being ejected out of 12, which is the anus. So we're kind of focusing on around here, the stomach, small intestine, large intestine, but that's the whole journey from start to finish. And I thought I'd mention this as well. The appendix, you saw it there. There's that's what it looks like. It just kind of sticks out down that area, so at kind of the end of the large intestine, and that's what it looks like in a jar. That is not mine, but that is an example of one. And it's been unknown really why it's there, but the kind of most recent evidence suggests it's basically where gut bacteria are stored. So what happens if you're ill, then the gut bacteria end up getting flushed out of the body. If you've got diarrhea or whatever, or some norovirus or whatever. So if that happens, we're talking about good bacteria here in inverted commas. This is bacteria which helps you break down food. So what they think the appendix does is it then allows the bacteria to be restored should it be lost. So a safe house for gut bacteria. So what is going on in this picture? So the appendix we know something about. If we go back a long time ago, it really wasn't known at all what the function of the stomach and the intestine and so on was. But this guy called Alexis St. Martin inadvertently helped move science forward, because what happened to him was he was accidentally shot and it left a hole in his stomach. And this is what this guy here is putting some sort of device into. So what a doctor decided to do, I guess with Alexis St. Martin's permission, was dangled food on a rope into his stomach to investigate digestion, and found that bits of food got broken down and also that there was acid in the stomach. So certain things that we took for granted, but this guy somehow managed to survive with a hole in his stomach, and yeah, it led to moving things forward in terms of knowledge of the digestive system. So thank you to Alexis St. Martin for accidentally being shot in the stomach. Something also to mention is just food's journey. Again, this is, whenever we do this, it kind of surprises people. So food is about 5 hours in the stomach, then it's about 7 hours in the small intestine, then it's got up to 3 days in the large intestine, and what that means is, as long as you're well, Thursday morning's breakfast is Saturday afternoon's poo. So even your breakfast, if you have your breakfast at 8 o'clock in the morning, 8 o'clock at night, it's still, it's probably just coming to the end of the small intestine before heading to the large intestine. The large intestine, the job is to remove sort of water from your poo. Talking of poo, I'll just leave that there. This is a sort of public health slide. So just hopefully you're in the correct place on this. This is the Bristol stool chart. So if you're not in the correct place, you might want to consider some dietary alterations. So that's the kind of basics and kind of assumed knowledge that you should have about the digestive system. And then what's actually in the course is a feature of the small intestine, which is villi, villi, and a bit like alveoli, villi is plural, villus is the singular. So, and within the villi, you've got capillaries and lacteals, so you want to be able to say what they do as well. So here's our digestive system, and we're just kind of focusing on this area here. So the problem is that when you eat food, you can't, a crisp, for example, you're not going to be able to get that directly into your blood. It's not going to go. It's too large. It's not going to be soluble. So what's going to happen is it's got to be broken down. So This process, as I said before, starts in the mouth with an enzyme called amylase, and what has to happen is these foods have to be digested to smaller soluble molecules which are able to move from your gut into your blood. So some of these you might know. So starch is potatoes, chips, rice, many things, and here it is on the left hand side. So a large molecule too big to get into the blood, so it is broken down to glucose and maltose. You can see that they are smaller. Protein, hopefully you know this from the reverse process when we looked at DNA. DNA is the code for something, and then these are put together. A chain of these makes a protein, and then the reverse happens during digestion. So the protein is broken down to amino acids. Okay, so protein broken down to amino acids. So it's a bit of a strange thing if you think about if you're eating meat. What's happened is the DNA within the animal has been, the chicken or whatever, has been used to make amino acids, which have then been joined together to make the protein. You then eat the protein in of the chicken breast or whatever, then your body breaks it back down to amino acids, and then your body uses the amino acids again to make your protein. Okay, so I think this is kind of going back and forward, but in digestion, protein is broken down to amino acids. And then this one, which you may not be familiar with, this is fat here, and you can see it's made up of two components. So this here, the blue one, and then the red one, and you've got an enzyme which breaks it down to these two components, and they are glycerol and fatty acids. Okay, so this is the glycerol here, and then you've got 3 fatty acids produced. So these are the products of digestion. So have a look at this YouTube film. It's from TED. So search for how your digestive system works. So we're going to focus now on the small intestine. So we're assuming that before we've got to the small intestine, in the mouth and in the stomach, those processes we talked about before have occurred. Starch, protein, fats have been broken down, and they're now ready to be taken into the bloodstream. They're smaller, they're soluble. And if you were to look within the small intestine, this is what you have. So we mentioned it before. This is the villi, and this is an actual image from someone or something's stomach. So they look like little fingers. So there's a villi there, there's another one there, there's another one there. So they're finger-like projections, and that's where absorption occurs. And here's another kind of focusing in on one. So imagine this area in white, this is the inside of your small intestine, and then here you've got the blood vessels and something called a lacteal. Okay, so we talked about glucose, amino acids, fatty acids, and glycerol, and there's a bit of separation here as to what happens. So here's your villi. Now the glucose and amino acids, they go into the blood. So they go into the blood capillaries here. So imagine them moving across. The fatty acids and the glycerol, on the other hand, they go into this structure here called the lacteal. Okay, so fatty acids and glycerol into lacteals in the middle, and then the glucose and amino acids go into the blood capillary, into the bloodstream directly. Here's another image of them. So similar to alveoli, for villi we talked about various features which allow them to be efficient at their function, and villi have the same. And as we go through these, you'll see, if you can remember the features of alveoli, you're going to remember the features of villi as well. Again, there's a large number of them. They're all packed in, you can see. So you've got a large surface area. What would that be? Thin walls, again, to allow the nutrients to move across. And again, you can see it here, a good blood supply. Okay, so villi, alveoli, exactly the same kind of features which allow substances to move across efficiently. For alveoli, we're looking at gases. For villi, we're looking at the products of digestion. So here's something I'd like to do. So draw this table, and this just gives you a comparison of alveoli and villi. So draw this table and tick or cross this in each box. Press pause and do that. So this is what you should have. Alveoli are in the lungs, villi are not. Alveoli are not in the small intestine, villi are. This next one's been put in to trick you. So alveoli, we know that they don't contain lacteals. However, I'm hoping you put a cross for this one as well, because lacteals don't absorb glucose and amino acids, do they? They absorb fatty acids and glycerol. So that was a bit cheeky, but that should be a cross. And both alveoli and villi have got those three features: thin walls, large surface area, and a good blood supply. So here's an exam paper question, so have a go at this. The following diagram shows a cross-section of some villi in the small intestine. Explain why the structure and number of villi make absorption an efficient process in the small intestine. Press pause and have a go. And this is what you should have. So you can pause that and before I speak. So you should have two of those features. You get 2 marks if you say thin wall, large surface area and good blood supply. And then the final mark is for saying that there are a large number of villi, which increases surface area. Okay, so it has to be something, you have to say something about the large number. Okay, have a break. Okay, so the next part of this we're just going to do is go back over revision of alveoli and villi, and then have a look at back through what we've covered in Unit 2 as well, and there'll be a couple of wee tasks related to that. So press pause, put down numbers 1 to 4, see if you can write down the answers. So number 1, starch is broken down to maltose or glucose. You might remember SAM, starch, amylase, maltose. Protein, number 2, is broken down to amino acids. Fat, 3 and 4, is broken down to fatty acids and glycerol. Okay, next, excuse me, here's our absorption in the lungs and small intestine. So put down numbers 5 to 10, and I want you to say what each of the correct answers is. So basically the gases and the products of digestion which are going into out of the alveoli and into the villi. Press pause. So 5, the gas which is going into the alveoli is CO2 or carbon dioxide. 6, the important gas we need, which is going into the blood, is oxygen. Then 7 and 8, moving from the villi and moving into the villi capillaries, that is glucose and amino acids going in there. 9 and 10, going into the lacteal, is fatty acids and glycerol. Okay, so you can actually going to have to miss out a couple of these, but you can have a go at this. You miss out heterozygous, xylem, and lymphocyte, but let's have a try. And this is all things we've been doing recently, so see if you can write down the definition of each. Press pause and do that. So the equator is the middle of the cell, and that is where chromosomes line up during mitosis. So we covered that during cell division. Stem cell is an unspecialized cell, and it can copy itself, and it's involved in growth and repair. Cerebellum, part of the brain, can you remember what happens there? So that is the part of the brain which maintains balance and coordinates movement. Then you've got synapse, so that is a gap between a neuron. So this is from the control and communication topic, and chemicals transfer messages across them. Glucagon is a hormone, and it's released by the pancreas when blood glucose levels are low. And you might have also said what its function is, and its function is to turn glycogen into glucose at the liver. Diploid is a type of cell, and that is a cell with two sets of chromosomes, e.g. skin cells. Heterozygous down to lymphocyte. If you've covered this, you might have had a go. That's what you should have for those. And finally, villi is finger-like projections in the small intestine, and nutrients from the food are absorbed into their capillaries and lacteals. And well done if you mention specifically what goes into capillaries and what goes into lacteals. So just to finish off, I'm going to give you some problem-solving questions related to Unit 2 material. So just as you go through these, just put down the number, press pause, and I'll give you the answer. So number 1, the table below shows the results of an investigation into the effect of temperature on egg laying in adult red spider mites. So you've got 3 different temperatures, and then the average length of egg laying period per days, and then the average number of eggs laid per female during the egg laying period. So as the temperature increases, the average number of eggs laid per female per day what? So the kind of important thing is per day. So what you have to do is a little sum, because it's telling you how many eggs are laid in the whole period. But there's a different number of days for each one. So the first one you have to do, I'm rubbish at drawing right with this, 72, needs one of those graphics tablets. So you'd have to do 72 over 24 to work out how many eggs were laid per day, and that'll give you an answer there. Similar for this, 72 over 18, and then 72 over 12. So when you do the wee sums, what you get is the number increases. So I guess you can sort of see it from the 72 over 12 is going to be the largest number. Then there's number 2, press pause. The diploid number of chromosomes in a cell from a kangaroo is 12. Which line in the table below identifies the number of chromosomes for the cell type shown? So a diploid is 12. So that means I'm going to attempt to draw here again. This is supposed to be sperm. So if the diploid is 12, then the sperm is going to be 6 because it's haploid. That is an egg. It's going to be 6 as well. So sperm 12, that's wrong, must be 6. Skin 6, no, because skin is going to be diploid. Nerve is diploid too. Remember, everything except from sperm and egg is diploid, has 2 sets of chromosomes. Zygote, that is the fertilized egg. So it's going to have, that's when the 6 and the 6 join together. So that's right. So 2 is D. 3, press pause. So the chart below shows the percentage of men and women with obesity at different ages in a population. So men, the lighter colour, and there's the percentage within that age group, and then you've got the different age groups. Women, the darker colour. Which of the following statements is true? For each age group, there is a higher percentage of obese men than obese women. Well, that's not right, is it? Because look at this one. There's more women than men. For each age group, there is a higher percentage of obese women than obese men. That's not right. In fact, you can pick out that one where men is higher than women. Obesity in men and women increases with age up to 64. Let's see, so men increasing, women increasing, yep, that's an increase, so that might be okay. Obesity in men and women decreases with age up to 64. Well, that's not right because it'd be going like this. So it looks like it's C, and that's correct. 4, press pause. During a reflex action, the speed at which information flows was measured to be 90 meters per second. Calculate how long it would take for the information to complete a reflex arc which was 0.9 meters in length. So if we're saying that it goes 90, oh, my drawing is a bit better now. I'm getting used to this. 90 in 1 second. It's like I'm back at primary school here. 90 in 1 second. How long would it take to do 0.9 meters? So if it's 90 in 1 second to get to 0.9 meters, then 90 divided by 100 is 0.9. So you do the same to 1 second, you divide by 100, and that gives you an answer of 0.01. Okay, so does that make sense? So 90 meters in 1 second is what it travels. So that means if it was traveling 9 meters, it would be 0.1 seconds. So if it was 0.9 meters, it would be 0.01 seconds. 5, 6, 7, this is related to chocolate. Press pause and do this. What we have here is a label from a bar of chocolate with the nutritional information in 100 grams, then in a bar, and then as a percentage of what you should be eating on a daily basis. And then we've got a table which is specifically about salt, and then 3 questions. So using information from table 1 and 2, identify the salt category to which this chocolate bar belongs. So salt content, and it's given you per 100 grams here. So salt is 0.4. So 0.4 grams is going to be medium. Okay. As part number 6, as shown in table 1, saturated fat makes up part of the total fat of this chocolate bar. So we are looking at this. So calculate the percentage of total fat which is saturated. So total fat which is saturated, so you're looking at these two values. So the total fat's 25 and 20 of it is saturated. So it's 20 over 25 as a percentage, which is 80%. Part 2, 1 bar of this chocolate contains 630 kilojoules, which is 7.5% of the reference intake. Calculate the total number of kilojoules which should be consumed daily. So if 630 is equal to 7.5%, then you have to get to 100%. So it's 6, the answer is 8,400 by the way. So it's 630 divided by 7.5. Oh dear. 630 over 7.5. And that would give you 1%, and you want to get to 100%. So 630 times 7.5 times 100, which gives you 8,400. 8. Each skin cell in a mouse has 40 chromosomes. How many chromosomes were present in each cell after dividing 4 times during cell culture? Press pause and do that. This is a bit of a trick question. So this should be C. So it's asking you about the number of chromosomes. So the number of chromosomes isn't going to change. Every time your cells divide, you don't have an increase in the number of chromosomes. So they should remain the same. If it was asking you about the number of cells, you'd have a bigger number there, but the number of chromosomes remains the same. So 8 is C. Number 9, press pause. The table below shows the blood glucose levels of 2 people after eating the same meal, and it tells you that the normal range of blood glucose levels is 82 to 110. It's milligrams per, I think that's deciliter, not really important, the units, but the time after eating the meal here, and then you've got the blood glucose levels. So using the information given, which of the following statements is correct? Person A always stayed within the normal range. So did they stay within 82 and 110? No. Person B was out with normal range 180 minutes after eating. That looks right, it was 160. But let's check the others. Person B had a level twice as high as that after of person A, 180 minutes after eating. 90 times 2 is 180, so that's not right. And then D, person A and B both had their highest levels 90 minutes after eating. Well, B did, but A didn't. Number 10, press pause. Regular physical activity can help reduce the risk of heart disease. The table shows the percentage of males and females of different age groups who meet the weekly recommendations for physical activity. So again, it's picking out an incorrect statement. So which of the following statements is not correct? The percentage of males meeting the weekly recommendations always decreases as age increases. Always decreases as age increases. Yep, it's always going down. B, the percentage of males always decreases. Down, up, down, down. So that looks like it could be, B might be wrong. That looks like it might be the answer because it does go back up, doesn't it? 26% of males aged 35 to 44 don't meet the weekly recommendations. Yep, 100 minus 74 is 26. 35% of females aged 25 to 34 don't. Yep, 100 minus 65 is 35. So, as suspected, correct answer is B. Number 11, do this one. The table shows some information about causes of adult deaths in Scotland. We've got cancer, coronary heart disease, and chronic obstructive pulmonary disease. Calculate the simple whole number ratio of male deaths to female deaths due to coronary heart disease. Important word here is simple. You've got to simplify it. So coronary heart disease, we're focusing on this, and it is male to female. So it's 165 to 105, and then you can simplify that, and that gives you 11 to 7. Okay. So that is if you divide both sides by 15, isn't it? And there's 12 and 13. Press pause, try these. People with type 1 diabetes need to inject insulin. The table contains information about some of the different types of insulin. So, for example, type B takes an hour to start working. There's no peak in insulin levels, and it's in the blood for 20 to 26 hours. And then you've got the one down here. So S, for example, is works faster, but isn't in the blood for so long. So a fast-acting type of insulin can be injected just before meals. Identify the type of insulin that's best suited for this. So which one is fast-acting? That's S, because it's working within 15 minutes. Another type of insulin can be injected once a day to provide a steady supply of insulin to the body. Which would be the most effective at doing this? So once a day would be the one that encompasses 24 hours. So that would be P. And then our last one, as part of a study, so press pause. As part of a study into the health of a group of students, blood glucose readings were taken over a period of time. The graph shows the readings for one of the students. So there's your blood glucose concentration here, and there's the day progressing from 7 a.m. to 9 p.m. Here's normal blood glucose, which you can see should remain steady, but the student's blood glucose, you can see it's more variable. So 14, at what time did the student's blood glucose reach its maximum value? So the highest value is going to be here. So that was 1900 hours. 15, calculate the percentage decrease in blood glucose between 9 o'clock and 11 o'clock. So 9 o'clock is here and 11 o'clock is here. So what you need to do is work out what these two values are. So we're looking here and we're looking here. Oh, that's not very good, but we're going from, what is that? There's 1, 2, 3, 4, 5 boxes between 4 and 5. So that means that each box must be 0.2. So we have gone from 4.2 to 6. So to do percentage change, you do the change over the original times 100. So you're doing the change is 1.8 divided by the original, which is 4.2. So it's 1.8 over 4.2 times 100. So it has gone down by 30%. That's how you do that.